DNA replication. Deoxyribonucleic acid, or DNA, is the carrier of genetic information in all living creatures. Before a cell divides, it replicates its DNA in the chromosomes, producing another identical DNA. DNA replication begins with an enzyme called helicase. Helicase starts by breaking the hydrogen bond between adenine and thymine of the DNA, forming a replication fork. As it continues to break the bonds along the entire length of the molecule, the two double strands separate from each other. A protein called topoisomerase moves between the strands. Several single-stranded binding proteins attach to the strands of the separated DNA on the outside. Both proteins prevent the two strands from curling back. When DNA strands are separated, an enzyme called primase synthesizes complementary ribonucleic acid, or RNA, nucleotides called RNA primers randomly on both the strands in the opposite directions. After a few RNA nucleotides are placed on the strands, an enzyme complex called DNA polymerase continues to synthesize new complementary DNA strands from the 5' end to the 3' end. Matching nucleotides are added one at a time. One of the strands is continuously synthesized and called the leading strand. The other strand is synthesized in short pieces and called the lagging strand. The short stretches of DNA that make up the lagging strand are known as Okazaki fragments. Another enzyme, RNase H, removes the RNA primers that began the DNA strand synthesis. An enzyme, DNA ligase, links short stretches of DNA together to create one long continuous DNA strand. The topoisomerase and single-stranded binding proteins move away, leaving two new copies of the original DNA molecule. Summary: DNA replication produces two exact copies of the original molecule. The new DNA double helix will have one new strand and one original strand.